Dear students, in this lecture we will discuss about the heme proteins. This is the part third of heme proteins. In the previous lecture, we have discussed about the heme proteins introduction and cytochrome P450, that is a heme protein. And in this lecture, we will discuss about the peroxidase and catalase. So, peroxidase and catalase are the enzymes. They are heme based enzymes. So, these are also under the category heme proteins. So, again, the peroxidase and catalase, they are heme containing enzymes. Both enzymes have high spin ferric heme group at the active site. So, iron third is the active site of the heme group of this heme protein. These two heme proteins. So peroxidase is also a heme protein and catalase is also a heme protein. Now, what is the function of these enzymes? So the enzymes, they protect the living systems from high concentrations of hydrogen peroxides. So they dissociates the hydrogen peroxide, which is dangerous for the living systems. Both acts on the hydrogen peroxides, but in the different way. So we will study how they can uh, they can uh, f- uh, they can act on the hydrogen peroxides if the hydrogen peroxide is present in the biological system. So if we see the peroxidases where they found, what are the different peroxidases? So horseradish peroxidase, ligninase, and manganese peroxidase are plant-based peroxidases. So these are the peroxidases that are present in the plants. While this lactoperoxidase, myeloperoxidase, then salivary peroxidase, thyroid peroxidase, they are human heme peroxidases. So these peroxidases, they are found in the human beings, while these are the peroxidases which are found in the plants. So the peroxidases are present in the plants as well as in the human beings. So if we see the heme peroxidases, they can found, if we divide it into subfamilies, then we will find that peroxidases that are present in bacteria, in fungi, and in plants. This is one of the subfamily, and the other subfamily is the animals. So these can be divided into these subfamilies, or they can also divided into some major classes. So the class 1 is intracellular peroxidases. So these are the peroxidases, they are present in the intracellular parts. Yeast cyclochrome C peroxidase. This is a soluble protein. Okay, because we know that peroxidase is a heme protein. So this cytochrome C peroxidase is an example of intracellular peroxidase, which is a soluble protein, and it is found in the mitochondrial electron transport chain. Okay, so there it protects uh, the uh, the part that the particular mitochondrial transport chain from the toxic peroxides. Okay, so these peroxides, they may affect the electron transport chain. Thus this kind of peroxidase, it protects the system from the toxic peroxides because they disintegrate, they react upon the hydrogen peroxides. Like the other peroxide, that is intracellular peroxide, is ascorbate peroxidase, and that causes the removal of hydrogen peroxide in chloroplast and cytosols in the higher plants. Okay, so these are the this is the class uh, one class that affects the H2O2, the its protection of the tissues from the toxicity of peroxides. This is the second class of peroxidase, which is secretory fungal peroxidases. So the examples are lignase and manganese dependent peroxidases. These are the monomeric glycoproteins that causes the degradation of lignin. So this is the second major class of the peroxidase enzyme. The third class of peroxidase is secretory plant peroxidase. These are again the monomeric glycoproteins that can perform multiple functions in the tissues, like they can remove hydrogen peroxide from chloroplast and cytosol. They can oxidize toxic compounds. They are also involved in some biosynthesis of the cell wall, and they are also acts as defense mechanism towards the wounding. So they are responsible for defense of the wounds. So these are the three classes of peroxidase enzyme. Now, if we see the action of peroxidase, then this peroxidase uses hydrogen peroxide as an electron acceptor and catalyze the other substances. So, like in this reaction, you can see that hydrogen peroxide can reduce to H2O so that it will oxidize the other substrate. So generally, in case of peroxidase enzyme, the H2O2 converts into H2O. Generally, the conversion of H2O2 to H2O is a two electron oxidation reduction reaction in which H2O get reduced to water and enzyme get oxidized. So this is a kind of redox reaction in which H2O will reduce to water and enzyme will get oxidized. And if we see the catalase enzyme, then catalase enzymes, they are found in blood, bone marrow, kidney, liver, and mucous membrane. So almost all parts of the body covers the catalase enzyme. And what will catalase enzyme do with the hydrogen peroxide? Hydrogen peroxide will disproportionate in presence of catalase enzyme. So actually the catalase enzyme causes disproportionation of H2O2 into H2O and O2. It means H2O2 at one side it is reduced to H2O and on the another side it get oxidized to O2, and thus it protects the cell of the living systems from oxidative damage by reactive oxygen species that are also called as the free radicals. So they are helpful in reducing the activity of free radicals. Now come to the structure of peroxidase and catalase. So if we see the structure of peroxidase and catalase, then we will find that in case of peroxidase, the iron center is having +3 oxidation state. The 4 coordination, they are satisfied by the 4 nitrogen of the 4 pyrrole rings, while the fifth ligand is the histidine residue of the protein of this peroxidase, while the sixth ligand is the water molecule. If we see the case of catalase, then we again find that the metal center is again +3 iron center and it is attached to 4 nitrogen. The fifth ligand is the tyrosine of the protein that surrounds this heme prosthetic group, and the sixth coordination is satisfied by this H2O molecule. And if you see this red line, actually this is the protein that surrounds this heme, and this tyrosine is a residue of this protein. Likewise, in case of peroxidase also, this is the surrounding is the protein, and this histidine residue is the part of this protein. So the only difference in the structure of catalase and peroxidase is that in case of peroxidase, the fifth ligand is the histidine residue, while in case of catalase, the fifth ligand is the tyrosine residue of the protein. Now we will see the reactions of the peroxidases. What are the different reactions of the peroxidases? The peroxidase shows, I have already told you that the peroxidase generally reduces H2O2 to H2O. So this is a kind of redox reaction where enzyme get oxidized and the peroxide get reduced. So this is the general reaction that is catalyzed by the peroxidase, where you will find this H2O2 on reduction, it converts into H2O. So here you will find the enzyme. This is the peroxidase enzyme. It can easily bind H2O2. This H2O2 can replace the water molecule that is present on the sixth coordination position of this iron center, and after binding H2O2, this H2O can evolve, and this enzyme get oxidized to this compound 1, in which you can see the iron has the 4 oxidation state, and then this enzyme can easily reduced to the enzyme by protonation, and then it can converts into H2O. I will show you the mechanism of its reaction. So this figure is showing the mechanism of peroxidases. This is the peroxidase enzyme. This is the mechanism of the peroxide enzyme, that is cytochrome C peroxidase. So here you can see this is the enzyme. L is the histidine in case of peroxidase. The active site of the enzyme is the histidine part of the protein, histidine residue, and the arginine or asparagine of the side chains of the proteins. And these amino acid residues, they are responsible for the heterolytic cleavage of this oxygen-oxygen bond. Okay, so for the breaking of this oxygen oxygen bond, this will require the charge separation, positive and negative charge separation. Okay, so the histidine and asparagine, they perform this kind of role, and the basic imidazole uh, group that is present in this histidine, it helps in the proton transfer. So this is the proton, so it helps in the proton transfer to the oxygen atom that is departing. So this is the oxygen atom which is departing. So this histidine imidazole group, it facilitates the transfer of this proton to the departing oxygen, so that H2O molecule can easily be evolved. So during this reaction, H2O will evolve, and this will convert into compound 1, which is an organic cation radical. And in this case, you can see the iron is in its 4 oxidation state. Okay, so this 4 oxidation state with this double bond O, it is called as ferril. Okay, so this is ferril, where you will find that this the oxygen atom is either protonated or linked to a donor group via hydrogen bond. So this is the linked with any donor atom with a hydrogen bond. So this is the unstable species, and this can easily this can easily get reduced. So this will get reduced through protonation, and during this protonation, it converts this oxygen to H2O molecule, and reduce itself to the peroxidase enzyme, so that the peroxidase enzyme can regenerate easily. Now, this is the catalytic cycle of catalase enzyme. So this catalase enzyme, this generally disproportionates H2O to H2O and O2. It means it get reduced to H2O and it get oxidized to O2. And the catalytic cycle of this catalase enzyme, it can be It occurs in two steps. In the first step, this is the first step, okay? So in the first step, this enzyme will binds to this H2O2 again, like the peroxidase, where the histidine and arginine, they helps in the heterolysis of this oxygen-oxygen bond, and imidazole group of this histidine will facilitate the proton transfer to this departing oxygen. This L is tyrosine here, while in case of peroxidase, it is histidine. So that after removal of water molecule, this will convert into compound 1 again, which is having the 4 oxidation state of iron. This compound 1 can oxidize various kind of molecules like H2O2, nitrite, formate, and ethanol. Okay, so this is actually highly oxidized, so it wants to reduce. So it get reduced to the catalase enzyme, and during this reaction it will disproportionate this H2O to H2O and O2. So this is the catalytic cycle of this catalase enzyme. This is the second step. While in the first step, the enzyme will get oxidized and one H2O molecule will reduce to H2O. In the second step, this compound one will again get reduced to the enzyme, and here the disproportionation of H2O into H2O and O2 will takes place. So this is all about the peroxidase and catalase enzyme. In this lecture, we have discussed about their position, their location in the living system. We had classified the peroxidase enzyme into three classes and two subfamilies. Then we had studied the different reactions that are catalyzed by peroxidase and catalase. The peroxidase, they reduce the H2O2 to H2O, while the catalase, in case of catalase, H2O2 get disproportionated into H2O and O2. The peroxide, in both the cases, the water molecule expelled due to the two amino acid residues that are arginine and histidine. So they facilitates the evolution of H2O molecule, then conversion of the enzyme into the compound 1, that is the oxidation of enzyme to compound 1, in which the iron center was in 4 plus oxidation state, and this 4 plus oxidation state, it can easily oxidize to many of the molecules. So in case of catalase, this complex, it can disproportionate H2O2 to H2O and O2. So this is all about peroxidase and catalase.